it's my pleasure to introduce James Michielini, a Pop Bio grad student giving his QE seminar. I first met James in 2018, in January 2018, when he showed up to take my statistics course at Tufts. And then at the end of the quarter, someone encouraged him to ask me about research, and I had a visiting scientist from China who was planning to do research at 2 sites about 400 miles apart from each other, and I realized wasn't eligible for a driver's license. So I said to James, do you like to drive? And he stuck around and did a senior thesis, which he published in Ecology Letters, which is one of the top journals in ecology. He ended up becoming the lead author on the Visiting Chinese Scholars paper and publishing that as a postbac. He followed me here to UC Davis, and it's delightful to have him. And I guess one other anecdote about his first year, 2018, is that I was actually visiting here, collaborating with Neil Williams that summer, and they developed this system where James, who just had the stats class, would field questions from the other undergrads working on the project and answer them in a first round, and then we'd all do Zoom meetings every Wednesday afternoon where he would, or maybe at that time it was Skype, I don't remember, but anyway, where he would organize them and give them preliminary advice so we could meet and do it. And it was, that just speaks both to his excellence as a scientist, but also his patience and interest. He's always also sought out opportunities to teach, especially quantitative stuff, to biologists and ecologists, both of which are traits I really admire. So
thanks,
now you'll hear about what he's doing now.
All right. Thank you, Elizabeth. Thanks, everybody, for being here. I'm excited to tell you a little bit about my research for my dissertation, exploring the consequences of urbanization for butterflies across different levels of biological organization. And so, let's get started here. Yeah, urbanization itself is a phenomenon where people tend to concentrate in small locations in cities. And in the past century, the proportion of people living in cities in the United States has more than doubled. As this concentration of humans has increased in cities, we see lots of transformation to the landscape as well. Many more impervious surfaces and other threats to wildlife coming from things like pollution and other factors which make things potentially inhospitable for species that might have lived in these locations prior. And so we often think that cities have relatively low biodiversity or lower species richness, which is the case for many vertebrate taxa where this has been studied. For example, in birds, we can look at this result from a meta-analysis in 2017, which shows that relative to suburban sites, urban sites have about half the species that are present in suburban sites, and rural and natural sites tend to have about the same, if not more. But this isn't always the case for all taxa. For some insects, and for insect pollinators in particular, these patterns don't always hold. A recent study that was published just this month out of Germany found that vacant lots that were overgrown with weeds and wildflowers, what they call urban wastelands, weren't actually worse off when compared to natural areas. And if we look at the results here, the urban wastelands are grayed out and the natural sites, these protected areas, are in white. You can see that for the number of wild bee species, if anything, urban sites tend to accommodate more bee species on average. This is also the case in another study that was published a few years ago in Bees and Hoverflies, which show that cities, when compared to natural protected areas, tended to have more, if not the same, species richness. But as you all know, I'm interested in butterflies, and most studies of butterfly species, so richness is actually negatively affected by urbanization. About 3/4 of the studies documenting butterfly species richness across the urban-rural gradient finds declines in richness with increasing levels of urbanization. But we know relatively little about the underlying mechanisms that drive this pattern, and there is a lot of interest in pollinator gardening and conservation in urban green spaces to support insect pollinators like butterflies. So my interest lies in sort of understanding a little bit more about these mechanisms. And for my dissertation, I'm interested in looking at these patterns at a few different spatial or biological scales to understand how they might play out or how these patterns build up. My first chapter explores biodiversity trends across an urban-rural gradient and explores species urban affinity. My second chapter focuses on exploring the demographic effects of urbanization for a common urban butterfly. That's the pipevine swallowtail, which we have here in California. And my last chapter will be understanding how habitat context affects movement behavior at road edges. And for the purpose of this talk, just for time, I'll mostly be focusing on the first two chapters. And so the first question of my first chapter is, how does species richness change in relation to urbanization? You've seen it play out in a few other studies. I was interested in this system in Massachusetts, where we have some wonderful data from the Massachusetts Butterfly Club. So this is a group that I was formerly a part of back when I was at Tufts that organizes trips to see butterflies throughout the state. And they report all butterfly species that they observe in a particular area. And thus, we can infer from those reports which species are present at a particular site and which species are absent. And I'll just note that this community is really, really great at identifying individuals to species. They're much better at identifying butterflies that are hard to sort of distinguish than I am. That's just one of the benefits of working with this kind of data and this excellent group. This data set includes records that go back about 3 decades to 1991. And they have 35,000 trips worth of data. So 35,000 outings on which they are going out to survey butterflies on the landscape. And because they report all species that they observed, we infer that species absences where they are not found, as I said. And so I'm interested in understanding how species richness, we can look at species richness using these data. And so I'm using incidence-based metrics to estimate total richness of each town in Massachusetts. We're interested in looking at the township level because that is the narrowest geographic resolution we can look at these data. Sometimes only the information of the town where the survey occurred is what is provided. And often there are privacy concerns not only for the people who are reporting potentially from their backyards, but also for the potential of folks to go out and collect butterflies to extinction, which is something that has happened in the past in some instances. And so, as I said, I'm using incidence-based metrics. I'm specifically using the Chow estimator, which you can think of like a species accumulation curve. It gives a similar result. Essentially, what it takes is the number of species that you observe in a particular location, and then also the ratio of singleton species, so species you only observe once at a particular site, to doubleton species, species that you observe twice in a particular site or town in this instance, to infer what species may not have been found there. So it gives us an estimate of species richness with some associated error. So that's great. We have that for all of our towns. Now, how do we measure urbanization? When we say urbanization, we can sort of easily conceptualize. We can differentiate between like a city and a rural area, but it's hard to know which variables are best at delineating between the two. And this is true even in the literature. So this is a pie chart is from a review that was published in 2019, showing all of the different ways people categorize urbanness or differentiate along the urban-rural gradient. A lot of people use structural forms of the landscape, so how much greenness there is, or impervious structure there is, the grayness. And I figured I'll look at a bunch of them. I want to see how they relate to each other before deciding what is best to look at in terms of classifying how it might affect butterfly species richness. And so the first data set that I looked at was the proportion of land developed from the National Land Cover Database. This is provided by the US Geologic Survey. And what this data layer gives is it splits the entire United States up into 30 square meter grids and assigns each of those pixels on the landscape as belonging to one of a few different land cover categories. And what I did was I calculated for each town in Massachusetts what proportion of those pixels within the town are considered developed in some way. And so this includes everything from parks and lawns on the low intensity development end, all the way up to like parking lots and physical like office buildings on the high end. I also looked at the proportion of land that's just impervious surface, so much more looking at what portion of the land is dominated by asphalt or concrete or other surfaces that prevent water from infiltrating the surface. I looked at greenness using the Enhanced Vegetation Index from MODIS imagery, and this is just a measure of reflectance of green light from the earth. And I averaged for each town what that measure is for a particular location. So if we look at this picture in particular, zoomed in on Boston, you can see that Boston itself would have a lower greenness score on average than its surrounding towns or other parts of the state. I also looked at artificial light at night, which is a measure of sky glow, the amount of light just sort of atmospheric hanging out in the sky at any given point overnight, and calculated that the same way, just calculated an average score from this index for each town. And then lastly, I just took the population density of each town as well, which is easy enough. And so interested in how are all of these variables correlated with each other, and it turns out quite highly, all of them. The lowest correlation we found is 0.68, which for an ecologist is a little striking to see. We call that strong. And this is a parallelogram showing all of the pairwise correlations between each two variables. And you can see that none of them are colored white or sort of near that spectrum. They're all sort of high intensity blue, which means they're positively correlated with each other. For example, we see greenness is, we see impervious surface, for example, is highly correlated with development. Or red, in this case, greenness was negatively correlated with all of the other variables, which makes sort of intuitive sense. And so looking at all of these different variables on a principal components analysis, we see that things fall out really cleanly. Our first principal component explains 90% of the variance among all of our townships. And we can easily distinguish with that on the left side here, those rural sites from the most urban towns. The reason things look squished here is because these axes are relative to the proportion of variance explained, and the second principal component only explains about 7 to 9%. This is what it looks like when we map out all of the towns on that principal component, and to my estimation, these seem like a very good estimate of urbanization, so I'm going to be using this as our predictor for urbanization in the model. I used a generalized additive model to explore the relationship between these two variables. Generalized additive models, or GAMs, are really nice because they are flexible and allow you to interpret potential nonlinear trends between two variables. And it does this by generating a spline, or like this squiggly curve, that is penalized by the data itself to make sure that it's not overfitting. If you're familiar with GAMs, this is essentially the model that I ran, and these are the results that we got. Overall, well, I'll first just explain what we're seeing here. So on the x-axis is that urbanization, that first principal component of all of our urban metric variables, and then on the y-axis is the species richness of any particular town. Each town is represented by a single dot with associated errors, and the size of the points represent how many trips the Massachusetts Butterfly Club actually took in that particular town. And so we see a lot of variation between towns, and overall, we can see there is a slight decline in richness with increasing urbanization, which conforms with what other previous studies have found. But we also see this peak around this median urbanization, which is interesting. If we look at a representative town, this is Leominster in Massachusetts. It has a state forest as part of it. It has a bunch of development, some single-family homes, a nice downtown. It's near an airport, as well as like a right-of-way and golf course. And this sort of area is what we predict as having the highest species richness from this particular model. Interestingly, Boston also had relatively high species richness, more so than other highly urbanized towns. And that was interesting to us as well. So in terms of that intermediate peak, for most of the way we rationalize this, or what our interpretation of this result is that the climax community for most of Massachusetts in the most rural parts of the state is not necessarily ideal for butterflies. So if we allow succession to occur all the way to a climax community, Massachusetts tends to be dominated by tall hardwood forests with very little herbaceous cover on the ground. And that herbaceous cover and the wildflowers that grow there are actually quite meaningful for the butterflies. And so in our lowest urbanized places, we only have those butterfly specialists that are able to live in the highly forested locations. Butterflies might much more prefer to live in places like this, where we have regular mowing, where there is conventional agriculture, not sort of large-scale industrial agriculture, monocultures that use a lot of glyphosate or other pesticides, but rather have a lot of opportunities for various spontaneous forbs and host plants and wildflowers to pop up to support them as resources. And interestingly, we found that even urban towns tend to have some pockets of suitable habitat. So Boston, for example, popped up as having somewhat high species richness. And part of that we can attribute to a lot of green space that actually occurs in the city of Boston. This, for example, is a picture of Arnold Arboretum, which I think this was taken in the fall. But if you go in the spring, it's covered in wildflowers. There are a lot of different and diverse communities of plants that are there. So how does species community composition change across the urban-rural gradient in Massachusetts? I'm interested in sort of digging a little bit deeper to know sort of, are the same species being represented in these urban and rural locations? Is there turnover from the rural to urban sites? And more specifically, do we find any urban exploiter butterflies? We know that, and this is going to be like the second question of my first chapter. We know that urban affinity varies a lot from species to species, where studies of vertebrates classify species in their response to urbanization in 3 categories. The first are urban avoiders, which see generally monotonic declines in abundance under any development. One example of this is the bobolink, which requires intact wild meadows. Any increase in the amount of urbanization in or around its habitat tends to drive this species away. We also have tolerators, which is another class that people categorize different species into, where minimal changes in abundance occur across the range of urban development. So they're capable of coping with some level of human presence. These are occasionally called adapters or suburban adapters, like the swallow, for example. And then lastly, we have our urban exploiters, which are species that tend to increase in abundance with urban development or human population. And an extreme and intuitive example in this instance are pigeons or the rock dove. But this also includes other species like house sparrows and raccoons, species that we often associate with urban living. And if we look at the urban affinities of these different birds, we can see that avoiders have negative urban affinities, tolerators have urban affinities around 0, whereas exploiters have positive urban affinities. And again, I'm going to be using the same data from the Massachusetts Butterfly Club to answer this question. And we have data for 89 different species. As well as and for those species, we have the presences and absences from trips that we're using to estimate urban affinity. And so let's just look at one example. This is the common ringlet, a lovely butterfly that we find in the state of Massachusetts. And I'm just showing you here a map of the proportion of trips on which this species was found by the club. We want to compare this to the degree of urbanization in each town. Here, I just have a map of that first principal component, all of our urban metrics. And what I'm doing for this analysis is using a generalized mixed model to predict the presence of this species on the landscape. So we're predicting presence and using urbanization as a predictor. In this model, we're also including region to account for the fact that sometimes we don't expect certain species to show up in a region simply because their range doesn't extend throughout the entire state. And we're also including a random effective town to account for unexplained or unrelated variation among towns that has nothing to do with urbanization, whether it's urban or not. And so to look at the results for this analysis, sorry, jumped forward. This species, as urbanization increases, the species tends to typically decline in presence. We see it less and less often on those trips made by the Massachusetts Butterfly Club in the most urbanized towns. So and because this relationship was significant, we would call this species an urban avoider. And so essentially, I did this analysis and I repeated it for all 89 species in our data set, and we get something that looks like this. This is a lot of information. You can ignore the species codes on the x-axis. All of these points represent individual species, and they are ranked sort of from the lowest urban affinity to the highest urban affinity. We see that the common ringlet comes out here on this plot where it has negative urban affinity, far below 0. It's our urban avoider. And overall, we saw 34 of our 89 species were classified as urban avoiders by this analysis. And interestingly, we saw no urban exploiters. So all other points on this plot, all these black dots, we would consider tolerators or no trend species, which is what I have right here. Interestingly, the species that popped up as having the highest urban affinity wasn't actually an exploiter. This is the cabbage white. You were probably all familiar with this butterfly. You've definitely seen it around Davis, present a lot in the agricultural fields around Davis as well. And this species is highly adaptable. It's present in cities a lot. But interestingly, and unlike something like pigeons, they are not present in cities at a higher abundance than they are in natural sites. They're not disproportionately or only found in cities. They're also found in very rural areas. And so for this reason, we call them tolerators. So that led us to think, so what butterflies do we end up seeing in the cities? Do city butterflies tend to have higher urban affinity relative to other species in the community? And so what we did was I selected the most urban towns across the state and explored the relationship between urban affinity and the relative abundance of that butterfly. And we would expect that butterflies that are most commonly found in the city have relatively higher urban affinities, right? But that actually is not what we ended up finding. So there are two species that pop out here. The cabbage white and the monarch are the most urban or most common urban butterflies. Those are the two all the way there on the right. But the general pattern that we observe is for all other butterfly species in our data set in Massachusetts, we actually see that a negative relationship between relative abundance and urban affinity. And this suggests that the other species present in high abundance in city aren't like doing well in cities per se, they're just generally very abundant species. They happen to spill over into urban communities. And so this is not the pattern that we observe in birds, just to reiterate, where the commonest species are almost always the urban exploiters. And the patterns of species richness that we saw in butterflies appear to be different than what we see in bees. So that was my first chapter. In my second chapter, I'm interested in diving a little bit deeper to explore the demographic effects of urbanization for a common urban butterfly, the pipevine swallowtail. And nicely enough, the pipevine swallowtail we have in Massachusetts, and happens to be a tolerator based on the analysis that we did, and is also present here in California. And so I'll be doing a bunch of field work in California to understand how they cope with the urban environment. And just a little bit of background about pipevine swallowtails. They are host plant specialists. They feed exclusively on the vine Aristolochia californica, or California Dutchman's pipe. They are found naturally in riverine areas that contain pipevine, and they typically emerge in spring with one large generation. They overwinter as pupae, so that chrysalis all the way on the bottom right. They will emerge in spring as adults. They will reproduce. They will lay their eggs on pipevine, which they will eat, often when they are young, gregariously, so in small groups. They will eat the plant together before dispersing. They develop, and then they become adults again and have about two or a few more, sometimes smaller, successive generations throughout the year. So you have this one large generation in the spring and then smaller ones that occur throughout the year. Interestingly, pipevine swallowtails are commonly found in cities. You can find them in Davis. They're in the Arboretum, as is pipevine. But they are one of the very few species where we have some sense of their demography in cities. So this one study in 2014 out of SF State found that lower survivorship in the Bay Area in urban gardens versus natural areas. This is pretty interesting and leads us to a mystery. So why are these butterflies super common in cities despite potentially having lower survivorship here? And I'm interested in understanding the differences in urban versus rural habitat, because it's unclear whether urban gardens are acting as population sinks or ecological traps. Are they attracting pipevine swallowtails here, even though they don't perform as well in cities? Or are these gardens potentially good? And I want to know sort of how the habitat context affects how these butterflies perform in these two settings. And so just to give a very gross oversimplification, here is an example of a natural habitat where you might find a pipevine swallowtail. This is in Mississippi Bar. You can see lots of different patches of pipevine growing up around each other. And then this is my very weedy garden in my backyard where I have a single pipevine plant that managed to successfully take hold. I want to highlight a couple things. The first thing is that the first difference between these two environments is that my backyard gets some supplemental watering. There's like a drip irrigation system which provides water to the plant throughout the year. And supplemental watering is a phenomenon that can affect phenology, or the timing of species and life history. Irrigation can prolong greenness by up to 2 months in urban areas, a previous study recently found. And a longer growing season generally just means that there's more available forage for late season generations. And so we're thinking perhaps urban gardens could act as a seasonal refuge, especially here in Northern California, where we tend to have a very arid, dry, late season summer. Is it possible that these urban gardens that have watered pipevine are sort of buffering the population or providing an extra resource at a time when they otherwise have very low quality resources? And so that is the first research question of my second chapter. Does host plant quality change over the growing season, and can supplemental watering improve the survival of late season caterpillars? This is work that I have yet to do, so if you have any feedback for me, I'd love to hear it. And the general methods that I'm looking at doing here for this observational study are to visit gardened and natural pipevine plants around the Sacramento and Davis area. And where I find eggs and young larvae, I want to revisit those sites at high frequency to track the survivorship of those developing juveniles. I'm looking to survey like about 10 natural pipevines, 10 gardens that are unwatered, and 10 gardens that are watered. And I expect to find results that look somewhat like these across our different treatments. Not really an experiment, but. In our first generation, I expect to see that in natural settings, there is slightly higher survivorship of those juveniles compared to urban gardens, watered or unwatered, in part because these plants are not water stressed at the time in nature. But as time goes on, I expect to see, as host plant quality in the natural sites tends to decline, as those plants start to senesce and desiccate due to aridity, that the survivorship of pipevine and urban gardens will tend to stay the same, or at least not decrease as much over time, potentially suggesting an interaction between the time of year and this treatment. In addition to that, I'll measure leaf water potential and trichome density, a few of these other plant quality metrics to try to get a sense of how that changes over the season and whether they might be important for the survivorship of the butterflies. I'm also interested in doing an experiment complementing that observational study, rearing pipevine swallowtail butterflies that come from urban and rural sites on plants that also come from rural and urban sites. And so this study is a factorial study of host plant origin and caterpillar origin, where I monitor the survival of these butterflies that are coming from these different places with different plants to see whether or not the natural habitat has better or worse plant quality for the developing butterflies. And also looking to redo this for each generation to understand whether survivorship changes over the season are similar to those I might see in the field. And from this, I want to get a sense of whether host plant quality differs between sites and over time. Another factor I'm interested in understanding more about in terms of the habitat context for these butterflies is the isolation of host plants in urban gardens. Another thing you can tell is different between these two different sites is that I only have a single plant, whereas in the natural settings, there are often large patches of plants that sort of scattered throughout the landscape. And these could prove to be very important for the butterfly from the caterpillar context and from their perspective. So a study in the 90s found that plant size tends not to influence the number of eggs that a female oviposits on a particular plant. So even on very small plants, she will lay very many eggs, as eggs are typically laid in large clusters. And we know that caterpillars can often defoliate plants. This is true for studies of pipevine swallowtails that were done in Texas, and also just generally from our understanding of butterflies from childhood. And so it's possible that caterpillars, which often sort of wander away from their natal plant as they get larger and eat more host plant, could run into this problem where they're running out of food. And so I just want to note very simply, could caterpillars run out of food in isolated isolated gardens? Which is the second question of my second chapter. So do we see density-dependent survival in small host plant patches? And are urban patches sufficient for developing clusters of pipevine swallowtails to survive? I plan to explore this using an experiment where in the field I will have 3 treatments that vary in host plant availability. I will plant harvested pipevine in pots and place them in 3 different treatments, one that only has a single plant, one that has 5 plants, and then one with 10, and place 10 eggs belonging to a single brood. So these are all siblings belonging to a single mother in each of these treatments, and essentially just track their survival over time from eggs to larvae to pupae. Monitor the total leaf area and leaf damage, and examine the weight of the pupae, which is correlated to adult weight and in turn affects fecundity, or the number of eggs that a butterfly can produce. I expect that with more host plant available, we would expect to see higher survival and lower defoliation over time, so that as the number of host plants increases, we have higher survivorship. This would suggest if we find this pattern, that density dependence might limit the ability of pipevine swallowtails in some urban gardens from persisting. And so with these 2 factors, I'm interested in understanding how survivorship or habitat context affects pipevine swallowtails in urban settings, and what this might tell us about the population viability and population growth of these butterflies in urban environments. In this case, we expect that water to potentially be good for them, but that for isolation of host plant patches to be bad for them. And this could be important, communication strategy if we need to, tell gardeners, perhaps you want to plant multiple pipevine patches in your backyard, in your garden, if you want to support a more thriving population. And so that was the completion of my second chapter. And just to review, in my first chapter, I'm interested in characterizing the biodiversity trends across the urban-rural gradient of butterflies and characterizing those species' urban affinity. So far, I found that richness tends to decline with urbanization, but some urbanization seems to be okay for butterfly diversity. And unlike invertebrates, we tend not to find any urban exploiter butterflies, at least in our analysis. And in my second chapter, I'm interested in exploring the demographic effects of urbanization for a common urban butterfly to reveal whether aspects of host plant quality in terms of watering or quantity in terms of the patch size affect their demography and survivorship in cities. So I just want to thanks everyone for coming. Thank you to my committee members, Louis, Rick, Emily, Kate, and Jay. Thank you, Elizabeth, and all the members of the Crone Lab that are here at Davis and also the ones at Tufts. Thanks to CPB, my cohort, the seed grant group that we're working on also in pipevine swallowtails, the EVE office staff, as well as the insect ecology group, and all of the undergrads that help make this work possible. Thanks. Yeah, Lori.
I guess I'm curious about your second chapter. You're talking a lot about kind of within a single season, the effect of watering on survivorship.
Sure.
And I'm curious how you would expect that to look different across years, thinking about like some years are hotter and drier, and
Yep.
some years are less. And like, would you expect to see the same trends or further interactions between years as far as
That's a great question. I expect that in a drought year, for example, for these effects to be exacerbated, where depending on, of course, how people are watering their gardens, perhaps they're sort of like coming back. But If the treatments sort of remain the same from like one year to the next, I would expect, yeah, exacerbation of these particular effects, and potentially just smaller populations of butterflies. It depends, like from a year-to-year context, if drought is affecting the population, perhaps survivorship over 1 year might influence the amount of butterflies you see in the next year, and so survivorship might increase if it's sort of like 2 drought years connected to each other, but yeah, Peter.
I thought it was really cool that you had no urban affinity species. No
Yeah.
Species that, yeah. And you point out that that's different from vertebrates.
Mhm.
And I guess the question is there anything interesting about what causes the difference between groups that have affinity and those that don't?
Yeah, like different functional traits. That might, yeah. So yes, that is something I'm interested in looking at. I have done a sort of first stab preliminary analysis at understanding whether butterflies that have certain functional traits tend to perform better or worse in cities. And so I did this both just as a first pass with, and the models revealed that butterflies that tended to be, or butterflies that are smaller, butterflies that don't overwinter as larvae, ones that have fewer host plants, and one other thing. All of those traits are typically associated with higher urban affinity on average. But I also ran those models with a phylogenetic correction, or including effects of like a phylogeny, and it seems that you can't really parse out all of those effects of those particular traits end up dropping out when you include phylogeny. So it's hard to say sort of like, whether that's a property of the particular trait or whether that has something else to do with these clades. Does that answer your question?
I guess I'm wondering why mammals and birds, for example, have species that are
Oh, sure.
and why butterflies don't.
Yeah, it's hard to say. There are similar studies that have looked at this has been done in birds, looking at sort of which ones tend to be urban explorers, and often it's species that have higher brain to body size ratio, so or birds that are like smarter, and also that are more flexible social groups. For mammals, I'm not as sure. In part, I mean, we think if you think intuitively about it, it sometimes just has to do with like the resource use context. But yeah, those are traits that sort of have popped out. Yeah, Oshi.
When creating the urbanization metric,
Mhm.
supposed to do get that data from one type point or like over 10 years that it was.
Yeah, that's a very good question. So all of these come from one time point. I did want to know how one of these metrics changes over time, just to make sure that I wasn't biasing anything in terms of the data. And so I looked at the National Land Cover Database. They release about every 4 years intervals, and those were perfectly correlated. It was like a correlation of 1, if not like 0.99, where development, I mean, it did change over time in Massachusetts, but it changed relatively like uniformly across all towns, I guess you could say. So I'm not super worried about like a potential bias in like time difference, if that makes sense. Yeah, Kristen.
In your urbanization model, you had urbanization and region as
Mhm.
independent variables. Did you not find that there was a strong correlation between the two?
Yeah, so there is a bit of correlation between the two. With the second region is sort of like the Boston metro area. Part of the reason we included those in our species-specific models was because we couldn't explicitly account for spatial autocorrelation, and that somewhat helps because if Boston, for example, or the Boston metro area is on average relatively more urbanized, then that will sort of like take some of the variance, if that makes sense. We also looked at these patterns sort of across the different regions as well, and it seems sort of concordant between them. So, yeah.
That makes
exciting to see the results of your experiments. I was curious if you thought about like developmental timing with the urban, like both the urban pipeline, like the first experiment you showed and the second experiment of like the amount of plants available, just like time to get to pupa, if that varies ever, or if that like might vary in the urban, non-urban context.
Yes, that is certainly something I'm thinking about. Generally speaking, I'm sure you know this. Arthropods and invertebrates often develop much more quickly when it is warmer. So in the late season, they are achieving that point a lot faster. That's certainly something I could look at, time to development from egg to pupa. And that's something I've done previously with somewhat similar work in Massachusetts. Yeah.
One really quick question, which is just, are there California pipeline in, why are there so many swallowtails in Massachusetts?
Oh, sorry. So they only eat Dutchman's pipe, California Dutchman's pipe here in California. They will feed really on any species in the genus Aristolochia. And on the East Coast, there are two main host plants that they use. There's Aristolochia macrophylla, which is just like large leaf Dutchman's pipe, and Virginia snakewort. And so that's what they're eating on the East Coast, yeah.
Sorry, I have two other random little things. You'll probably figure this out very quickly based on how the plants grow in the pots, but for your experiment where you're looking at the availability of Aristolochia versus how many individuals are there, might be like plant number might not be super consistent compared to like, I don't know, feet of vine.
Total
Right?
foliage
Some
amount,
really
yeah.
big- Yeah, anyway, pots, I'm sure you'll have, hopefully you won't have to deal with that, not all these different plants. And
That would be great.
Same time, but-
Yeah, exactly, right?
I was just like thinking, well, in terms of growth plants, I was like, oh.
Yeah.
And then I just missed how you accounted in, I'm sure you did in the first look at urbanization versus richness, the species accumulations of curve and the differences in sampling effort in those different locations.
Yeah, so that's something that's tricky with this data set that we have to contend with. Part of the reason we chose the Chow species richness estimator is because I believe it has relaxed assumptions compared to a normal species accumulation curve. It's like a non-parametric version of it. To an extent, we sort of can't get around the fact that like they only sample a certain amount in one particular town versus another. But if we look across all of the towns in Massachusetts in terms of their development score, and then all of the trips in Massachusetts in terms of their development score, those histograms look very much the same. So they're sort of they're sort of sampling representatively across the gradient of urbanization, even if they're not sampling representatively across each town. Does that make sense?
Yeah.
Okay.
Cool.
Well, if there are no more questions, let's thank James again.
Thank you. Thanks, people on Zoom.
How's it, Nancy?